this video we will discuss about the VCAM overexpression in the pancreatic cancer cells patients, pancreatic cancer cell lines, and also the pancreatic cancer patients. So, and how this VCAM genes comes when yesterday we did the mRNA characterizations of the PANC1 cell lines and the PANC1 cell lines, which is co-cultured with the macrophages. So from there they find out that VCAM is overexpressed, and in the literature when they check that the overexpression of VCAM is not been studied in the pancreatic cancer cell lines. So they decided to check VCAM overexpression in the pancreatic cell lines and also the pancreatic cancer affected patients. So first they check the overexpression of the VCAM in the PANC1 and KPN2 cell lines in two conditions. First they co-culture them alone, and in second they co-culture them with TAM, that is tumor associated macrophages or M2 macrophages. And what they have found out is that when they co-culture the cells with the TAM, they see the high overexpression of this VCAM. And these things they have also validated using the Western blot, where they can see that VCAM expressions are high when they are co-cultured with the TAMs. And after validating that the expression of VCAM gets upregulated when it's come in contact with the tumor associated macrophages, they wanted to check whether this is the same case in the pancreatic patients or not. So they did the analysis of 134 pancreatic patients, where they check the pancreatic tissues of pancreatic cancer tumor tissues and also the non-tumor pancreatic non-tumor tissues. And what they have find out is that the VCAM expression is statistically significant in the tumors tissues compared to the non-tumor tissues. And after that this in the panel D is the relative expression of this VCAM is shown here, where they can see that the VCAM expressions are high in most of the cases compared to the very few cases where VCAM expressions are low. And this indicates the ratio of tumors versus non-tumors. So anything which is in positive indicates that VCAM expressions are higher, and anything which is negative indicates that VCAM expressions are lower. After validating that, from these results of the 134 patients, they validated the VCAM expressions are high in the pancreatic tumors. And after that they randomly take the 4 samples and to the Western blot to check the VCAM expression levels, and they find out that in all 4 randomly chosen samples they see that high VCAM expression levels compared to its counter non-tumor tissues. And so this is from the panel E. They validated that VCAM expressions. They validated the VCAM high expressions from Western blot too. So they use two techniques. First one is the RNA expression levels, and second one is the Western blot. And thereafter they wanted to check these VCAM expression levels are present in other pancreatic cell lines and compare them with normal pancreatic cell lines. So they let me repeat this statement. So they, after validating that the VCAM expressions are high in the pancreatic cancer patients also, they wanted to check whether the VCAM expressions are high in the other pancreatic cancer cell lines and compare it with the normal pancreatic cell lines, that is HPD6C7. So, and what they have find out that in all the pancreatic cell lines, the expression levels of VCAM are statistically significant on the higher side compared to its normal pancreatic cell lines. And these VCAM expressions are higher in the KPN2 and the PANC1 cell lines. And this thing they have validated using the Western blot also, where we can see that expression levels of the VCAM are higher in the KPN2 cell lines and the PANC1 cell lines. The difference from the normal pancreatic cell lines is the maximum. So in this figure, so this figure in conclusion presented a point that VCAM expression levels are VCAMs are overexpressed in the not only in the pancreatic cell lines but also in the pancreatic cancer affected patients. Not only whenever I say cell lines, I talked about this pancreatic cancer cell lines, not the normal pancreatic cell lines. Whenever I want to talk about normal pancreatic cell lines, I specifically mention it. So I hope you understood how they have confirmed the high expression of the VCAM in the tumor cells and also in the pancreatic cancer affected patients. If you need to understand more about their experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.